Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Empowering Patient Care in Process and Quality Control Solutions for Cellular Therapies. I'm Roisin McGuigan, an editor at BioInsights, and joining me today are Christine Tishner and Simon Lenatz, who will discuss how to optimize point-of-care practices, focusing on analytical development and release testing. After the presentation today, we'll have a live Q&A session. We invite our audience to pose their questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. And if possible, please do indicate which speaker you'd like me to pose your question to. I'd also like to draw your attention to the Resources tab, which you can find on the right, where you can find more information on the topics covered today. Now, I'd like to introduce our presenters. Christine works at Miltenyi Biotech as a Senior Project Manager of Global Point-of-Care Strategy. She has extensive expertise in CAR T cell manufacturing and workflow planning for clinical studies. Simon is currently working on his PhD thesis at Cologne University Hospital, where he oversees the development of new CAR and TCR strategies to improve adoptive T-cell therapy. So without further ado, I'll now hand over to Christine, who will kick us off with the first presentation.
Thank you for the kind introduction and welcome everybody to today's webinar. I will start the first part of the webinar covering possibilities and challenges of point-of-care practice and introduce 3 success stories. Let me start with some fast facts about the development in the field of advanced therapies through the last decades. After the first successful bone marrow transplantation in the year 1956, it should still take many years until the first gene therapy trial started in the indication of metabolic disorders. One of the participants was Jesse Gelsinger, who died in 1999 as a reaction to the vector. As a consequence to his case, many clinical trials were set on hold until the new regulatory framework for cell and gene therapy was set in place in 2007-2008 in the EU and US respectively to standardize ATMP development. In 2011, Carl June and his medical team from the University of Pennsylvania were pioneering cancer therapy by, for the first time, applying CAR T cells to a leukemia patient. And many academic institutions were to follow this example the years after. In 2016, the first CGT named Strimvelis was approved to treat severe combined immune deficiency. And only 1 year later, but after a decades-long journey from technology proposal to finally finishing the clinical trial, the FDA approved Novartis Kymriah. Today, we see more than 40 approved CGTs on the market, and the field is still advancing, also based on new technologies like tumor-infiltrating lymphocytes and CRISPR-Cas-based CGTs to target solid tumors, for example, or sickle cell disease. On the other hand, the first approved CGT has already been withdrawn from the market for economic reasons, which also shows the limitation of these therapies. Miltenyi Biotech has been a strong partner in the development of cell and gene therapies for more than 30 years, providing breakthrough solutions based on an extensive product portfolio of more than 19,000 materials, devices, and consumables. To date, more than 150,000 CGT procedures are based on Miltenyi products and support the treatment of approximately 10,000 patients per year. CAR T-cell therapy has revolutionized cancer therapy, and similar potential can be expected in other indications, such as autoimmune disease. However, limitations currently remain since pharma development is long-lasting and expensive, and central manufacturing hubs need to handle complex manufacturing processes. Finally, market access and reimbursement can be very challenging depending on regions. Looking at the clinical perspective, treating physicians facing various constraints like commercial availability, high treatment cost, labor restriction, and long vein-to-vein times, just to name some of them. Today, point-of-care practice is being discussed as a potential solution to at least overcome some of the hurdles and broaden patients' access, especially in niche indications or outside the big markets, reduce cost associated with manufacturing, shorten the vein-to-vein time, or allowing higher involvement of treating physicians and researchers in academic institutions. I would like to use 3 real-world examples to explain the potential of point-of-care manufacturing in more detail and how they tackle the challenges of affordability in case of the Christian Medical College in Vellore in India, accessibility in case of the hospital clinic in Barcelona, Spain, and how academic research can be accelerated in case of the University Hospital Erlangen in Germany. Let's first look at India. With a population of more than 1.45 billion and a high incidence of B-cell malignancies, India is a big market. However, despite being an accepted standard of care for B-cell malignancies, access to CAR-T therapy cannot be considered as economically feasible. Decentralized manufacturing of anti-CD19 CAR-T was set up in an academic environment at Christian Medical College Vellore in context of the WellCARD preclinical study. One of the main drivers which makes this therapy unaffordable in low and middle income countries is the existing industry-driven centralized production. In 2023, Palani et al. Published that decentralized manufacturing of anti-CD19 CAR-Ts using a fully automated and closed system is feasible in a developing country setting. They described successful and robust manufacturing, meeting standard release criteria and showing a good safety profile in first clinical applications, and thus recommended in-hospital manufacturing for further clinical trials. The production cost per batch were approximately $35,000. To summarize the importance for the CGT landscape, decentralized manufacturing is suitable for CAR-T cell production in developing countries. And production costs generated in an academic non-profit setting can be a benchmark for pricing in low and middle income countries. But also in Europe, where approved CAR-T cell therapies are largely available, accessibility of treatment options is sometimes limited. Not so much due to high costs, but because of rare disease or off-label indications with unmet needs. The ARI project, which was initiated by scientists at the hospital clinic in Barcelona, is addressing this issue. The ARI project was initiated to deliver treatment options to all patients with B-cell malignancies, including off-label and rare disease. Therefore, they set up own lentiviral production in 2016. Based on that, their first clinical trial with CD19 CAR-Ts was started and obtained hospital exemption and reimbursement in 2021. They also obtained PRIME designation, and the therapy was accepted by EMEA in a pilot program for a centralized marketing authorization. Up on today, further clinical trials are ongoing for EU-wide approval and further indications. To summarize the importance for the CGT landscape, extensive use of hospital exemption for off-label and rare indications is possible, and so far more than 350 patients were treated in context of the ARI trials. Hospital Clinic Barcelona is the first public institution applying for marketing authorization. As mentioned already, CAR-T cell therapy is by now well established as standard of care in B-cell malignancies. Increasingly, CAR-T cells are now also being recognized in other indications, such as autoimmune disease, as the example of Erlangen will show. After 50 years of limited innovation in the treatment of SLE patients, research in this field has now become highly competitive. In 2021, Erlangen published first CAR-T infusion in SLE, which was well tolerated and led to drug-free remission of the patient. The year after, data of 5 treated SLE patients were published, and several companies like Bristol Myers Squibb, Cabaletta, Kyverna got interested and started their own preclinical trials. By 2023, all major CD19 CAR T-cell providers have started their own lupus trials. Until today, Erlangen has published data of 15 patients successfully treated, and further preclinical and clinical trials were initiated also with allogeneic CAR T cells. To summarize the importance for the CGT landscape, fast development of pioneering solutions thanks to academic research, and CAR T cells are a real game changer in treating SLE and other autoimmune diseases. However, despite the potential point-of-care manufacturing can offer, various challenges remain, like high requirements for the GMP infrastructure, limited scale-up capacities due to availability of premises in a hospital environment, access to devices, material and services in regions with limited local distribution, high requirement for quality control testing, and challenges connected with regulatory approval and reimbursement. From this list, which is certainly not yet complete, we want to take a closer look today at the support for clinical control analysis. I don't want to go into too much detail here, but if you look at the traditional CAR T-cell manufacturing workflow, which involves the cell preparation and cell selection of CAR T-cells from a leukapheresis product, followed by the cell activation and cell engineering before the expansion and final formulation of the drug product, it is clear that extensive analytical testing is needed. Focusing on the not safety relevant testings like purity, identity, content, and potency, we can support CGT customers by standardizing flow cytometry based on the MaxQuant analyzer. The MaxQuant operates on an audit-ready software compliant with 21 CFR Part 11. In addition, automated gating and analytics can be applied by using the so-called express modes. For multi-instrument alignment, we developed the Smart Gain technology. The whole is completed by a comprehensive portfolio of standardized reagents. Talking about standardized reagents, our StainExpress Dry Antibody Cocktail can standardize and accelerate your flow analysis, for example, for immune cell composition or transduction efficiency, for about 54% in time, and the schematic below shows how. This is the traditional workflow of sample preparation for flow analysis, which most of you are most likely familiar with, but I will briefly guide you through it. At first, you have to label your tube before compiling and adding all required reagents. Then you add your sample of choice and optional perform red cell lysis. Finally, you have to manually enter analytical settings before being ready to analyze. Whereas when using the StainExpress reagents, you only have to add the sample to the dried antibody cocktail in the tube and optionally perform red cell lysis. Since the tube comes with a barcode label, you can scan this and the MaxQuant analyzer choose appropriate settings automatically. This workflow not only significantly reduces time, but is also less error-prone and thus more robust. And finally, these are my key takeaways to wrap up on how point-of-care practice can empower patient care. At first, point-of-care or decentralized manufacturing can address urgent market needs, such as the reduction of vein-to-vein time, which is of very much importance for patients with rapidly progressing diseases, or the reduction of treatment costs, mainly applicable for countries with low incomes. Based on the experience of academic customers using our CAR-T manufacturing platform, Point-of-care setups can support overcoming current limitations, for instance, with respect to affordability and accessibility. And finally, standardized reagents like StainExpress can be the means of choice to meet stringent quality control testing requirements. And with that, I will hand over to Simon from University Hospital Cologne to provide some inspiring user insights.
Many thanks for a kind introduction and a warm welcome to all those interested in today's webinar. Today, I would like to talk about our CAR T-cell manufacturing process here at the University Hospital of Cologne. In my presentation, I would like to introduce you to our solutions for process and quality control for our CAR T-cell process. The idea of having our own CAR T-cell production facility here in Cologne came up a few years ago. However, the GMP unit was modified several times, but we were able to start the first test run supported by Miltenyi on the Prodigy platform in June 2022. Last year, we were able to finalize our SOPs and QC processes so that we started our first validation run in May and fulfilled the second validation run in August. In September last year, we had an audit and inspection by the Bezirksregierung Köln, which was very fruitful and showed us that we were on the right track. So we could start our next validation run in December, which was a 5-day run used for matrix validation. In the beginning of this year, we started our last validation run, and hopefully we will be able to achieve the manufacturing authorization for our homemade CAR T cells in mid of this year to treat our first patient. In order to achieve this great goal, we established a cross-functional team, which is similar to that in a company. With Professor Scheid as our head and manager of this company, lab and IT people, colleagues involved in QC management and the legal department, but also the facility management and Christine as our direct contact to Miltenyi Biotech. On the next slide, I would like to introduce you to our TCT process. Our 12-day CD19 CAR TCT process starts on day -1 by drawing a fresh leukapheresis from the patient. To determine if we have enough starting material to run the process, we include our first max quant-based in process control panel A. The actual TCT run starts on the next day by tubing set installation and priming. For the input parameter on the Prodigy machine, we run another immune cell composition panel before and after the CD4-CD8 enrichment process. The culture will be set up with 10 to the power of 8 CD3 positive cells and cultured with Tex-Mex with IL-7, IL-15 and human epi serum. The T cells will be activated by Transact, and on day 1, the pre-activated T cells will be transfused with the lentiviral vector. The process medium will be changed on day 5, serum-free medium, and QC sampling is done. The sample is analyzed by FACS for immune cell composition and transduction efficiency, as well as tested for mycoplasma and endotoxin. On day 9 to 11, a sample is sent to our external partner for mycoplasma testing, and the process ends on day 12 with the harvest and final formulation solution and full QC testing, also including visual inspection and vector copy number. Our FACS-based Panel A CAR T-cell immune cell composition is stained with a StainExpress cocktail and analyzed by the corresponding express mode. The panel can be used to analyze the percentage of the individual populations and to display and calculate the concentration. A distinction is made between T-cell subpopulations, but also between monocytes, B cells, neutrophils, eosinophils, and NK cells. On this slide, I would like to share exemplarily the raw data printout from our first TCT run after CD4 and CD8 enrichment. The gating will be performed automated by the express mode. At first, we can see a debris exclusion and gating for CD45 positive lymphocytes. After that, will be gated for living cells and CD3 T-cell subpopulations. Then will follow the gating for CD3 negative cells. On the right-hand side, you can see all concentrations and percentages for each individual cell type in a table. To determine transduction efficiency, we are using the StainExpress CAR T-cell transduction cocktail with CD19 CAR detection reagent, which is not included directly in the cocktail and have to be stained in advance. For the gating, we are also using the corresponding express mode. In panel B, a distinction is made between CAR-positive and CAR-negative T-cell subpopulations. Residual monocytes will be also analyzed. Here, I would like to share raw data printout from day 5. The gating was also performed automated by the express mode. The gating strategy is quite similar to panel A, except that now the proportion of CD4 and CD8 of CAR-positive and CAR-negative T cells is analyzed. And again, all concentrations and percentages are summarized in the table on the right-hand side. The sterility testing is done in-house using the BD-BACDEX system. Samples will be tested on day 5 from sampling and day 12 from the target cell bank. The testing is regulated by SOP and will be cultured under aerobe and anaerobe circumstances. Endotoxin testing is also regulated by SOP internally and will be checked for the same samples and tampons. For the testing, the EndoSafe system is used. For mycoplasma and vector copy number testing, an external service provider, Minerva, is used. Mycoplasma testing will be done on day 9 to 11 from sampling and day 12 from target cell bank. The sample will be analyzed by qPCR, including several controls on mycoplasma DNA targets. The vector copy number is determined from day 12 target cell bank. The vector copy number is calculated also based on qPCR. Vector copies per reference genes multiplied by transduction frequency results in number per transduced cell. From authority side, the VCN has to be smaller than 5. Both mycoplasma testing and vector copy number is regulated by our SOPs, and samples are sent to our GMP certified partner via express mail. We are using the enhanced feeding activity matrix, which is pre-installed on the Prodigy for our cultivation process. Process medium will be changed after feed and sampling on day 5. On this slide, I summarized the process output data of all runs we've made. To calculate the cell counts, the FACS data of panel A and B was used. All runs resulted in clinical relevant numbers of viable CAR T cells with strong robustness. The runs yielded in 4.5 to 6.5 by 10 to the power of 9 total T cells, with viability greater than 97%. The yield of CD19 CAR T cells was between 1.5 and 3 by 10 to the power of 9. At first, I have to note that all runs we've made were manufactured from different donor starting material. The following immune cell composition was analyzed with panel A before and after the CD4-CD8 enrichment on day 0, on day 5 and on day 12 of each individual run. They were summarized in the displayed dot plot. Additionally, the non-target cell bank was analyzed after the enrichment. In the first column, you can see the immune cell composition after the enrichment. Before the enrichment, the starting material contains a lot of T cells, depicted in green, but also a lot of other immune cells. After enrichment, there are mostly T cells in the positive fraction and some contaminating NKT cells, which are also get enriched via CD8 beads. In the non-target cell bank, on the other hand, there are almost exclusively monocytes, B cells and NK cells, and only a very few T cells. On day 5 and 12, the product contained almost exclusively T cells and only a smaller number of contaminating immune cells. The proportion of CD8 positive cells increases towards the end of the culture, which can be explained by IL-7 and IL-15 support. All our processes resulted in high transduction efficiencies, which was analyzed for each individual run on day 5 and on day 12. The transduction frequencies varied between 30 and 60% CAR-positive T cells. We further analyzed the CD4 to CD8 ratio of CAR T cells plotted on the right-hand side. At the end of each run, we checked the drug product for our release criteria. The CAR frequency needs to be higher than 10% in viable T cells. Viability of T cells needs to be greater than 80%. The purity of T cells need to be greater than 80% in lymphocytes. And the VCN has to be lower than 5. There's also a visual inspection of the drug product. As you can see in the table, we easily passed with all runs the release criteria. I have to note that we didn't measure the VCN for the test run, and run 4 was a 5-day run. At the end, I would like to sum this briefly up. 5 prodigy runs to manufacture anti-CD19 CAR T cells under GMP conditions were performed at the GMP manufacturing site here in Cologne. With the help of Max Quant flow panels, internal and external QC and in-process controls, GMP-compliant testing of the manufacturing process was able to meet the expectations of the authorities. All runs we carried out were able to pass the QC requirements and our release criteria. For us in Cologne, time is now, and hopefully we can achieve the granting of the manufacturing authorization this year. And as I'm more involved in CAR T-cell research, I would be very happy to see our self-developed anti-CD19 CTLA-4 switch CAR T-cell construct, which is already in the production pipeline, entering the clinic. And with this, I would like to end my presentation. I hope I could give you a good overview of our production process and all QC and in-process controls we implemented. Thank you very much for your attention, and